Hi friends, and today we will talk about mutations, type of mutations, and the consequences formed in human body by mutations. We will start with the mutation forms. There are mainly two factors which are causing mutations, that are spontaneous mutations and external factors. Spontaneously mutations are mutations that are caused spontaneously or that are caused in a point in a genes that in a genes or DNA that causes amino acid replacement or mutations of amino acid, which causes ultimately spontaneously mutations. And in external factors, chemical and radiations are two main factors affecting our genetics or DNA that causes mutations. And there could be other chromosomal mutations, point mutations, that chromosomal mutations, reconstitution of chromosomes. Mainly mutations occur either spontaneously, that I explained you. The following two factors of mutations should be distinguished. These are chromosomal mutations, reconstitutions of chromosomes, observable under the microscope, and point mutations. These point mutations are or replacement or gene mutations. The former can change in the supramolecular structures, which is the chromosomal mutations. This can change the supermolecular structure, and the point mutation can change in the nucleotide sequence in DNA, and accordingly in mRNA. Both in DNA and mRNA see the consequences in point mutations. And there are mainly four types of point mutations. This could be missense mutation, nonsense mutations, frame shift, and followed by silent mutations. So firstly, I will explain you the missense mutations. Mutations loading to replacement of nucleotide in codons and altering the sense of the codon. For an example, this alters the sense of codon. If it is AUG and CAU, then it will be changed, or it will be exchanged, and this alteration will sense this altered sense of codon, or this alteration will cause missense mutation. Now we'll talk about nonsense mutations. These nonsense mutations, in which a normal codon that specifies an amino acid is changed to one of the three terminal codons. This could be UAA, UAG, UGA start codon. So such mutations are known as nonsense mutations. This lead to the termination of the protein chain, and especially detrim- detrimental. These affects the mental situation of the patient. And the frame shift mutation, which result in a deletion or an insertion of a nucleotide, uh, they are so called because they shift the reading frame of the mRNA triplet codon. And the last one is a silent mutation, the replacement in codon leading to the codon degenerated together with the original one. The original codon and the replaced codon both are degraded, and this is the process which is called nonsense mutation. Normal codon is changed under this process, under this situation. So spontaneous gene mutation are derived by errors, and are followed in DNA replication that arise because of the thermal motion of atoms and molecules. Now, if we will talk about semi-conversative synthesis, then DNA loop appears under this situation. And if I explain you more precisely, then the insertion of a nucleoid that is not compulsory to the template nucleoid. This leads to the deletion or replacement of a nucleoid, usually lead to the formation of a loop in the DNA. The DNA, the double helix structure. The double helix structure, which somehow looks like this. If I show you in a small diagram here, and the another strand forming another base pairing strand here, and here the amino acids are attached to each other. So these are replaced. And if I if we talk about the semi-conservative synthesis, then here the DNA loop disappears. These loops disappears, and the formation of loop may be accompanied due to the formation of a pair different from a Watson-Crick pair due to the ability of nitrogen base to form unusual hydrogen bonds, and also due to tautomerism. And now we will talk about another situation on based on Vogel and Koppen analyzed 1293 amino acid replacements. So, if we talk about different point mutations, and these have been probabilities of based on the Vogel and Koppen 1977 analysis on which this is a scientific theory based on the 1293 amino acid replacements in phylogenetically linked proteins, which are accounted for by single replacements of nucleotide XY in codon XYZ. And furthermore, influential things said by Vogel and Kupan are explained by mutation. And here, if in conclusion, if I have to say about mutation, then this can be told that mutation, we have considered missense mutation which lead to the replacement of many amino acids, and the values of the probabilities of mutation in codons are variable thousand in numbers, and thousand type of mutations may occur, but these are the main points I have explained now in my research work. Thank you.